blood cells. Okay, so we have our erythrocytes. These are red blood cells. They carry oxygen and carbon dioxide to the blood. We have platelets. These create blood clots. Okay, so they're involved in immunity, but not as much. Okay, what we're really talking about is these guys, the leukocytes, right? They are divided into granulocytes. That's these three, which we're primarily going to talk about. And agrulocytes, that's these guys. Well, really, we're just going to talk about this guy next time. These four are going to be involved in innate immunity. Here they are in a blood smear, in case you ever need to look at them. Okay, so your defensive blood components. So your lymphocytes basically are involved. There's two types of agrulocytes. That's these guys. Your lymphocyte and your monocyte. Your lymphocyte is almost exclusively just this guy is adaptive immunity. We will talk about lymphocyte next time. They are natural killer lymphocytes. They are badasses. Here they are, lymphocytes. We will talk about them next time. That's just exciting. Monocytes, this guy. They leave the blood and mature into macrophages. The phagocytotic cells devour foreign objects. So these guys, they're in the army. They're in the army, the innate immunity army. We'll talk about them. Okay, here they are. This is what they look like in a blood smear. Okay, also, if you get a lab, you're going to get a white blood cell count lab, and that's really going to count your leukocytes. It's going to tell you if you have allergies or a parasitic worm infection, or if you have a bacterial disease, or if you have a viral infection. Okay, let's talk about phagocytosis. We talked about this a little bit in the overview video. Okay, so phagocytosis. This is phagocytosis here. It's going to just eat it, just engulf it, and pull it into itself. Any cell that's capable of phagocytosis is called a phagocyte. How do we come up with these names? Phagocytosis is not, we don't understand every little piece of it, but we pretty much know that the chemotaxis of phagocytes will say, hey, we need to eat this. Then it's going to adhere, so you can see all this here. It's going to adhere here. Then it's going to ingest the microbe. Then it's going to sort of digest it, if you will. It's going to digest it into a bunch of little pieces, and then it's going to spit it out. Also, it's going to take pieces of it through the Golgi, and then it's going to come back and sort of post it on its social media platform. Okay? So you've got chemotaxis, which tells it the way to go. You've got adhesion, which connects it. Then you have ingestion. Then you have maturation. Then you have basically the killing of the microbe here, and then you have elimination where it sticks it out. Okay? Okay, we're going to watch some videos on phagocytosis.
Phagocytosis is the ingestion of a solid, such as a microorganism or cellular debris, by a eukaryotic cell. Phagocytosis is used by some protozoans for obtaining nutrients, but this process is also used by certain cells of the immune system to fight infection. These immune system cells include macrophages and neutrophils, and are collectively called phagocytes.
Okay, next video.
Chemotaxis is the movement of a cell toward or away from a chemical stimulus. Phagocytes use pseudopods to move toward microorganisms or damaged cells at the site of infection, often arriving only minutes after infection. Chemotactic chemicals that attract phagocytes include microbial products, components of damaged cells, chemicals released by other white blood cells, and peptides derived from the complement system. The plasma membrane of phagocytes usually attaches to glycoproteins on the microorganism's surface. This process is called adherence. Adherence is made easier through opsonization, a process in which antibodies or complement proteins from the host coat the microbe's surface, serving as handles so that the phagocyte can more easily attach to the microorganism. The proteins that coat the microbe are called opsonins. The pseudopods fuse, forming a sac called a phagosome.
That is cool.
The phagosome enters the cytoplasm, where it fuses with lysosomes that contain digestive enzymes and other antimicrobial substances. The fused structure is called a phagolysosome. Digestion of most bacteria is complete within 10 to 30 minutes. After phagocytosis, the remaining debris is eliminated from the cell by exocytosis, in which the phagolysosome fuses with the plasma membrane and expels its contents. Some of the contents of the phagolysosome may also be presented on the cell membrane through a process called antigen processing and presentation.
Yep, do you see that? There's its social media going, hey, I ate something, right? We talked about that.
Let's review the steps in phagocytosis. The pseudopods of a phagocyte use chemotaxis to approach the microorganism. The pseudopods adhere to opsonins that are attached to the phagocyte. The microbe is ingested within a phagosome. The phagosome fuses with a lysosome to form a phagolysosome, in which digestive enzymes break apart the microorganism. Finally, the remaining debris is eliminated by exocytosis. Parts of the microorganism are presented on the surface of the phagosome.
Okay. Hopefully everyone feels reasonable about phagocytosis. Yes. Okay, part of the things that are happening here is you have toll-like receptors. TLRs, toll-like receptors. Okay, put myself down here. Okay, and they are integral membrane proteins produced by phagocytotic cells, and they bind to PAMPs, pathogen-associated molecular patterns. PAMPs. Pathogen. Let's say protein. Pathogen-associated molecular patterns. PAMPs. These initiate a defensive response. This is not a self-cell, not at all. When you see this, you need to kill it. This defensive response is a secretion of inflammatory mediators. Oh, remember, we talked about inflammation. So it's going to inflame. It's going to inflame things. It's going to stimulate an adaptive immune response. So it's going to I'm going to remember you. I'm going to remember you. If you come back next time, I will know. And apoptosis, right? It's going to degrade it and then poke holes in it, right? It's going to die. Okay? All right. So here's some toll-like receptors. They're just different ones. Here's peptidoglycan. Yes. Here's some things that are on bacteria that are not in you that would trigger this. Okay. Non-specific chemical defense against pathogens. You have interferons. These are proteins released by host cells to non-specifically inhibit the spread of viral infections. There's alpha and gamma ones. Here are the actions of them. Again, you're seeing that it's a virus. You're posting it here on your social media, and this is going to activate a series of reactions. Complement. Okay. Complement is a set of proteins designated numerically according to their order of discovery, and there is a lot of them. We are not going to go over all 25. There is a lot. Complement activation results in lysis, or death, of a foreign cell. It indirectly triggers inflammation and fever. Inflammation, separate from fever. And complement can be activated in 3 ways: a classical pathway, an alternative pathway, and a lectin. Remember, did we talk about lectins? Yes, we did. So we are going to do one more video just on complement, and then we should be done.